Esta conferencia se realizará en inglés. Good afternoon, ladies and gentlemen. Welcome to the 5th meeting of Age Open Science. It is a pleasure to have you, as it is not common that science is brought to the streets and placed on the spotlight for citizens to gain knowledge about a matter that concerns many, longevity. Allow me to give a big thank you for the audience that is also following online through our official website. To start, I call on Rogelio González, Professor of Medicine, Director of the Department of Medicine of the USAL and the Institute of Biomedical Research of Salamanca to take the floor. Thank you.
Good. Well, good morning. It is a pleasure for me to be here today in this Agustina Square of the Monterrey Palace and to have the fortune to be able to introduce Heinrich Jasper, Director of GENENTECH Inc. and Professor at the Bach Institute of Aging Research. He leads the biotechnology company dedicated to the research and application of innovative science to discover and develop drugs on the control of tissue regeneration and repair, metabolic homeostasis, and cell death. Heinrich has been researching aging and longevity for more than a decade, and his work was recognized with the Glenn Foundation Award in Biological Mechanisms of Aging Research in 2010. Heinrich Jasper comes to talk to us about a topic of concern to many of us, age-related stem cell dysfunction in barrier epithelia and intervention strategies. Please, Heinrich, you have the floor.
¿Me escuchan bien? ¿Se escucha? ¿No me escuchan todavía? ¿Ahora sí? ¿Se me está escuchando bien? Bueno, muchas gracias. Y quería agradecerle mucho a los organizadores de esta conferencia, que realmente es espectacular en este lugar. Y le agradezco por invitarnos a todos y darnos la oportunidad de hablar un poco de nuestro trabajo y divulgar nuestras observaciones del envejecimiento humano y el envejecimiento biológico general. I will speak in English because it's easier for me to talk about science in English. But I do want to really, again, thank everybody for coming and for really being interested in this very important topic that we hope will help us develop new therapies, new strategies for intervention in a wide range of chronic age-related diseases. So my laboratory is particularly interested in how aging impacts stem cell function. And stem cells, as many of you know, are really present in many of our tissues, and they maintain tissue function by regenerating the tissue after an injury. And you can imagine that as we age, tissues get damaged a lot. They're exposed to many different stressors, often to injury, and stem cells are called upon to be activated and produce new cells that ultimately act as functional cells for this tissue. Now, one important feature of aging is that the capacity of these cells to repair damage and to produce cells of that tissue decline. And this decline is something that we need to understand if we want to try to find interventions that improve tissue function and tissue homeostasis. So when I talk to people about stem cell function, I like to, no se avanza, a ver. Maybe I'll introduce another one, or this is it, right? Ah, está apagado. Bueno. Ahí sí. What I try to remind people is that somatic stem cells that are present throughout our tissues are cells that are really dangerous to some degree. These are cells that for a long period of time have to be maintained in a mitotically active state, so they have to have the capacity to produce new cells, to regenerate, to proliferate. But at the same time, they also have to maintain a certain plasticity to be able to generate many different cell types. And that feature of stem cells makes them very similar to cancer cells. Cancer cells have a certain plasticity and they certainly have the capacity to proliferate very strongly. So you can imagine that a human body that lives 70 years has to have the capacity to control the function of these cells very tightly. It has to be able to maintain the proliferative capacity without having them transform into cancer cells. And really, it's this balance between the capacity to regenerate and the potential for these cells to become deregulated and cause dysplastic or cancer phenotypes that we need to understand if we want to understand how homeostasis is maintained. Now, with age, these cells are exposed to the same stressors that I mentioned earlier, and they get damaged, and the control mechanisms that adjust their stem cell function, their proliferative capacity, also change and decline. So we need to understand how homeostasis is maintained. We need to identify ways of intervening to see whether we can modulate stem cell function in order to extend the health span of an individual or to even extend lifespan potentially. So my lab studies this process in a model system called Drosophila, and I'll talk about this in more detail. But the concept that we study in these model systems is universal to a degree in a variety of organisms, including humans. And the concept is that stem cells are, of course, not isolated in the tissue. They are maintained within a tissue microenvironment, which people call the niche. These are other cells that control the function of stem cells and their so-called quiescence until the tissue is damaged and the cells have to transition to an active state where they divide. They often self-renew. They produce new stem cells. And then they also go on to differentiate other cell types that are important for tissue function. We understand nowadays many mechanisms that actually control these processes. We know a lot about cell-autonomous regulators that control the stem cell state and the function of these stem cells. We understand that the niche, the cells surrounding these cells, also provide factors, proteins that control stem cell activity. And we understand that there is a systemic environment that, here we go, systemic factors that will actually control stem cell function from a distance. You can imagine that, for example, insulin, which is a very important growth factor, which is produced by beta cells in the pancreas, will actually influence stem cells in the brain, in the muscle, in other tissue types. So there is really a coordination throughout the body that control the function of these stem cells. Now, with age, all of these different things change. There are well-understood changes in the intrinsic function of the stem cells, which means these cells accumulate DNA damage, they accumulate damage in their proteome, and hence their intrinsic control mechanisms change. There are changes in the niche where a lot of the niche cells will die, will disappear, factors that they secrete will change, and accordingly, systemic changes will also influence the capacity of these stem cells to generate new cell types in the right way. So I want to talk about our work in Drosophila as a model system to understand these very basic mechanisms of how aging impacts stem cell function. And then I'll talk about a collaboration that we did with Juan Carlos Espizua Belmonte, who's been kind enough to invite us here and to coordinate this meeting, where we've explored how to engage a in vivo reprogramming strategy that is in principle being used to actually reprogram cells into stem cells, into pluripotent stem cells, as a way to improve tissue function in aging organisms. So now it moves too fast. Let's see if I can go back. Here we go. So as I mentioned, we use the fly as a model system. Drosophila has a long history in biological research, more than 100 years. People have used this organism to understand basic mechanisms of biology. And the reason is that these animals reproduce very fast. You can keep them in the lab. You can manipulate them genetically. And you can therefore understand how certain genes control certain functions of cells, of tissues. And accordingly, we can use it to understand which genes, which processes control aging of stem cells in this case, and how we can potentially tweak these, change them in order to improve tissue function. We study particularly stem cells of the intestine in the fly. The intestine, of course, is what the intestinal epithelium is what people call a barrier epithelium, because ultimately it's a barrier to the outside world. The fly ingests food, it ingests bacteria, it ingests yeasts, and all of this material, biological material, food, etc., toxins as well, will ultimately make impact on the barrier epithelium of the gut. And that barrier epithelium, therefore, needs to have defense mechanisms built in. The cells get damaged, they have to be replaced. The epithelial cells need to also respond to challenges and secrete certain factors that control bacteria, etc. And so you can imagine that this epithelium is under a lot of stress. It is lining the whole gastrointestinal tract of the fly. Here the stomach is actually in yellow, so you can see it's actually a normal gastrointestinal tract. You have a foregut, you have a stomach, you have a hindgut. When you do a cross-section through the hindgut, this is what you see. This is the epithelium I'm talking about. These are individual cells here, large enterocytes, which really are the workhorses of this tissue. They produce enzymes, they resorb nutrients, they fight bacteria, they interact with commensal bacteria, with good bacteria as well. And so these cells are the ones that get damaged and need to be replaced. And they're replaced by stem cells that are sitting under these enterocytes. And I outlined one here in a higher magnification. They're basically here between the epithelium and the basement membrane. And now I'm going backwards. Okay, here we go. And so if you draw a cartoon to highlight the features of this tissue, you can see that stem cells, again, are sitting on a visceral muscle layer. They will divide and give rise to what we call enteroblasts. These are non-differentiated cells that go on to then differentiate into two different cell types. Enterocytes, these large cells I mentioned, and a separate cell type called enteroendocrine cells, which are cells that produce hormones for the gut and therefore control gut behavior, they control immune responses, they control homeostasis of the tissue. In flies, we have a lot of ways of looking at these cells. We can label them in green here. You can see this is an image of a fly gut from the top, and you can see the stem cells here in green. You can see the large enterocytes. And we can label them, we can what we call lineage trace them. We can understand what the progenitor cells or the progeny of these cells are and how they differentiate. And because of this, we have a pretty good understanding for the mechanisms of regeneration here. So these stem cells, similar to all other stem cells I mentioned earlier, are mostly quiescent, become activated in response to stress, generate these enteroblasts that I mentioned earlier. There are interactions between the stem cells and the enteroblasts, and they go on to differentiate then with specific molecular, in response to specific molecular cues, they go on to differentiate into one of these other cell types. Now, this is a picture that's way too complicated to explain, but it just illustrates all the knowledge that the field has developed over the last 10 years to understand the different ways in which stem cells interact with the environment. These stem cells respond to signals from enterocytes, they respond to signals from the muscle, they respond to signals from blood cells, they respond to signals from insulin-producing cells, and we understand now what these signals are. They are very specific proteins that are being secreted by these other cells that control stem cell proliferation, function, activity, etc. What we've also learned is that in an aging fly, this whole system degenerates, and we go from, and this is very hard to see, unfortunately, in this screen, but these cells in a young animal are mostly individual, separated throughout, dispersed throughout the epithelium. In older animals, they become larger conglomerates of cells that are not properly differentiating, and maybe easier to see here in the next slide. Here we go. Where if you look at the cross-section, again, from a young animal, the tissue looks well organized, but in a cross-section from an old animal, you can see that on the basal side of the epithelium, these stem cells have started to over-proliferate and actually change the structure of the epithelium and develop a pathology that we could call dysplasia. So it's basically a precursor to cancerous growth. So the organization of this tissue is lost. And this is due to mechanisms that we've really tried to explore over the last 10 years or so. And I'll just give you an overview of the current model. What is happening in this tissue is that with age, the gastric region, the stomach that I pointed out earlier, becomes less able to secrete acids. So the acidity of the gut changes. That then leads to changes in the intestinal microbiome, because the bacteria in the fly will colonize the anterior part of the gut mostly. When the acidity of the gut changes, these same bacteria will start colonizing the posterior part of the gut, and that leads to inflammation. And that leads to basically a activation of inflammatory processes that then lead to the overactivation of stem cells because they respond to this challenge. They overproliferate, generate this dysplasia phenotype, and that then leads to changes in the function of the tissue. And I mentioned earlier, we're talking about the barrier epithelium that interacts with the environment, and it has to interact with the environment in a selective way. Only certain things can pass, other things have to stay out. So if you actually look at a fly that was feeding a blue food dye, and again, it's a bit hard to see here, in a young fly, this blue food dye will stay in the gut, so only the gut becomes blue. In an older fly, this same color penetrates the gut and goes into the whole animal. And so we call these flies Smurfs because you know the cartoon characters that are blue. You can see these blue flies when you have a population of old animals, you have blue flies in there, suggesting that the barrier function of this epithelium is disrupted. And actually, a lack of barrier function is something that we see in human patients of IBD, we see in human elderly population as well, as a marker of dysfunction of the intestinal barrier. Now, we've learned which mechanisms are driving this, and we found ways of perturbing these through genetic manipulations in the fly. And in these cases, we always saw lifespan extension. So really, it's the disruption of this barrier function that drives the increase in mortality with age in these animals. And so I will go through the next part a little bit faster, because I know I'm going to run out of time. But we've done a lot of work in recent years to try and understand what intrinsic age-related changes occur in the stem cells that contribute to this age-related dysplasia. And one of the pathways we've studied is called TOR. TOR is a signaling pathway that responds to nutrients. And every time a stem cell is activated to divide and regenerate the tissue, TOR is also activated. Now, TOR has two functions. It's important for the ability of stem cells to divide, but it's also important for stem cells to go into differentiation. And so what happens, every time you challenge the tissue, you will activate this TOR pathway, and you're increasing the probability of the stem cells to differentiate. And you can imagine if a stem cell differentiates without generating a new stem cell, you will lose your stem cell population over time. So the functional population of stem cells will decline over time in flies, and we actually tested whether this concept that we learned in the fly would also apply to a vertebrate system. We tested this in the mouse muscle and in the mouse airway epithelia, and it turns out that the same applies there. You have TOR signaling being activated in these other tissues in stem cells, and it contributes to the age-related loss of stem cell function. And we could show that a molecule called rapamycin, which is an inhibitor of TOR signaling, was actually able to restore function, the number of stem cells and the regenerative capacity of the epithelium, and hence we know that it also extends lifespan through a variety of mechanisms. Again, I'm going backwards. Another mechanism that we found is that stem cells have this interesting capacity to adjust to the environment, adjust the ability to divide either symmetrically or asymmetrically. And you can imagine that a stem cell that divides has a choice of generating two new stem cells, or to actually generate one new stem cell and a cell that goes on to differentiate. If you're generating two new stem cells, you can expand the pool of stem cells. You have more stem cells in the tissue. And that is important under certain contexts. That's why you can switch between symmetric divisions and asymmetric divisions. And there are specific mechanisms that control this. One is that where the cell divides, it has to divide its genome. And so there's a mechanism that allows chromosomes to be separated equally between neighboring cells. And it turns out that the angle with which the separation occurs will ultimately control whether you're generating a symmetric division or an asymmetric division. Now, in the old stem cells, most divisions are symmetric. And so you generate many more of these stem cells that then leads to this dysplasia phenotype I mentioned earlier. We also, again, a very complicated slide, but we also found that stem cells change their metabolism, and that the metabolism of an old stem cell resembles the metabolism of a cancer cell. There's a process in which cancer cells that are highly proliferative generate energy by a glycolytic mechanism that is less involved but faster, less efficient but faster than the mitochondrial oxidative metabolism. And it turns out that old stem cells have acquired properties that allow them to generate energy through this glycolytic mechanism. And that mechanism is very similar to transformed cells. You can actually induce oncogenic transformation in intestinal stem cells of the fly. And the metabolic mechanism is very similar. So in an old stem cells, they already acquire properties that are kind of similar to cancer cells, which also may explain why there is an age-related increase in the incidence of tumors in many of these tissues. The final story I want to just present briefly is the question of how do all these changes I described, how this change in stem cell behavior in the old gut influence the rest of the body. And we observed that the changes in the intestinal epithelium that are ultimately inflammatory changes are actually influencing the brain. And in this case, specific case, we've looked at the olfactory function of the animal. We know both in humans as well as in flies that the capacity to smell changes over time. And so there's an age-related decline in smell in humans, an age-related decline in smell in flies. You can measure it in different ways. And what we found is, and actually, let me just skip through this. What you also know, of course, is that infections like COVID, for example, impact your ability to smell. And what we found is that the changes in the gut that we observe with age are actually driving this decline in olfactory sensitivity. And this is due to a mechanism that has evolved to allow flies to stop eating when they have ingested a pathogen. So if they have a gastrointestinal infection, they will shut down the ability to smell because that prevents them from eating more. Now, in the aging gut, because of these changes I described earlier, you have this chronic inflammatory condition, and you have the secretion of cytokines in flies that are called UPDs, that we found impact the olfactory epithelium of the fly in the antennal lobes, and it will change the ability of the antennal lobe to sense these olfactory signals. This is, and I will skip through this briefly, this is just the evidence that this works, so that there's a signal from the gut affecting the olfactory epithelium. And we found the mechanism by which this occurs, which is that the signals from the gut impact glial cells, which are support cells of neurons, and change their metabolic activity. And they cannot support neurons in the right way. So I want to end then, as I said, with talking about our recent collaboration, if I can advance the slide, on in vivo reprogramming. Now, Juan Carlos's lab has actually shown a while ago that you can use a process of reprogramming in vivo to rejuvenate cells. And what we're doing there is we are basically changing the normal developmental path of a cell, which here is depicted in this landscape diagram. You can think of an embryonic cell up here. It goes down the valley of differentiation into certain states. It was always thought in developmental biology that that's an irreversible path, so that they just, from an egg, from an embryo, you go on to differentiate into all the different cell types of the organism, and you cannot go back. Well, it was shown by Shinya Yamanaka's lab that you can, in fact, go back. You can, by expressing very certain transcription factors, you can push cells back to this pluripotent state, or you can even have them jump from one state to another. So you can make a fibroblast into a neuron, for example, directly. And what Juan Carlos's lab has shown is that you can use that process in a very controlled fashion in vivo to try and rejuvenate cells, because this process of pushing them back all the way is ultimately a rejuvenation. But if you control it in the in vivo environment, you can get cells to rejuvenate partially, make them healthier that way, and not go all the way to a pluripotent stem cells, which would be a bad thing. There you could get teratomas. So that was shown by Juan Carlos's lab. It's been shown by other labs that you can take lung cells, you can partially reprogram them and put them back, and that will lead to a healthier tissue. Juan Carlos's lab has shown that that can be done in the skin as well. And David Sinclair's lab has shown that you can also do in vivo reprogramming in the eye, and you can change phenotypes that are associated with glaucoma. So there are a number of observations right now that suggest that these in vivo reprogramming approaches in certain disease contexts can be beneficial. And I'll skip through this. This is just a summary of Juan Carlos's previous work. But the question we wanted to address is whether this partial reprogramming is something that can be done not in a disease context, but in a healthy, aging animal. And we asked whether we could do this for long-term, using long-term treatment. And what we did there is basically took mice in which you can express these factors in a controlled fashion, in this case through the delivery of a molecule called doxycycline. You do this for 2 days, and then let the animal recover for 5 days, and you repeat this every week. And we asked whether you could do this for months. So can you actually put them on this cycling reprogramming protocol starting at 12 months of age, and then see what happens at 22 months of age? First question is, are they healthy? Do does this intervention become problematic over time? And it turns out that they seem very healthy, actually. They didn't really differ much in body weight or in survival or in neurological phenotypes. They didn't differ much from a normal wild-type animal. And then we asked whether we could see any changes in the tissue that are suggestive of a protection or a reduced aging phenotype. And I'm hoping I can move on here. And so we did a number of molecular assays, looking at transcription, for example. You can look at which genes are being transcribed more or less in these perturbed systems. Then ask, do we identify a pattern of gene expression that indicates certain biological changes in these tissues? And it turns out that, for example, a signature of inflammation, which is something you commonly see in aging tissues, that tissues express many genes that indicate a low-level inflammatory condition, that these were actually reversed. We could also show, with Steve Horvath's help, who is here as well, that the methylation patterns of various tissues, kidney, skin, for example, which change with age and are actually indicative of the progression of aging in these animals, could be pushed back as well. So in that molecular sense, these tissues were rejuvenated and seemed to be healthier. And we asked whether this would also apply to certain biological processes. We tested, in this case, skin repair. If you injure the skin, you know most of the wounds will heal. If you're young, they heal better and faster than if you're old. And it turns out that this is true as well in mice. It turns out that when we did this long-term reprogramming and then did an injury, the proliferation of stem cells in the skin, actually, which declines with age, was reversed back to an almost youthful level. The level of fibrosis that you see, which increases with age, is reduced to a youthful level, suggesting that the process of repair here has become more efficient, almost like a young animal. So really, it's a rejuvenation of these type of processes that are, again, driven by stem cells. And I want to end here. I want to thank, this is, of course, work that's done by many people. This is part of my lab here at GENENTECH. And I talked about work by all the people labeled in red here and our collaborators. And I want to thank you again so much for being here. And I think we have questions later after the next talk. Thank you. Thank you.
Thank you so much, Dr. Jasper, for sharing your investigations. To continue, Rogelio González will now introduce our next speaker.
Well, let me introduce now Cynthia Kenyon. She is the director of the Hillblom Center for the Biology of Aging at the USCSF. She is internationally recognized in the field of longevity, as she was one of the first people to study longevity and to conduct studies on the subject. The discovery by Kenyon and her colleagues that a single genome mutation could double the lifespan of C. elegans provoked an intensive study of the molecular biology of aging. Today, she will talk about the figure of physiology clocks for human aging. Cynthia, whenever you want.
Hi, everyone. It's fantastic to be here. I want to thank the organizers and Juan Carlos. It's such a beautiful place, so it's great. Let's see. May I have the first slide, please? Okay. So Juan Carlos encouraged me to talk about a little of the history of the field before I start talking about physiology clocks. So I'm going to try to do that. So first of all, just some thinking. So aging is kind of a universal condition. An old dog looks like a dog more than a person, but you can tell that it's old really easily. And that's true of most old animals. So there is something very characteristic about the changes that occur with aging, and that extends not to just these two species, but many others. But the rate of aging can vary. Those of you who heard Vera's talk, I think, got a great sense of that. But some animals age and die very quickly, like a mouse, or as you'll see, a fly, or a C. elegans worm. And some can live a really long time, including us, actually, but some even longer. So something is similar about aging, but it can happen at a different rate in different species. And this is really cool. If you can plot the chance that you will wake up in the morning and die that day, that is your mortality risk, over time as a species. And this is for humans, and what you see is that starting at about 30 years old, these are log plots here, you see there's a straight line here. So your chance of it dying goes up in a very predictable way. This is the human slope. A slope for a dog would be higher. A slope for a whale would be lower. But you can see that it starts really early. So there's something inside the person, I think, inside the person or the dog, even when it's young, that is dictating its rate of aging. It's somehow programmed, just in a sort of, I'm just saying that as a definition, to age at a certain rate. So what is that program? Okay, so how did these animals get these different lifespans? Genes changed during evolution. And that means there have to be genes that set the rate of aging, right? You don't have to do a single experiment, and you can figure that out. Okay, I just asked it to go forward, and it didn't. Okay, oops, now it went back. Hold on. Oh, the computer there. Okay, thank you, sorry. Okay, so now, this is interesting. I started working on aging in the early 1990s, a long time ago, and at that time, it was thought that aging wasn't controlled by anything. That it just happened. Your car does not really age. Well, it ages sort of, but it's not really there's not a program for aging. It just wears out. And that's what people thought happened for animals. You just wear out. There's no genes controlling it. And they had a reason to say that. They had a couple reasons, all of which I think were wrong. But anyway, I'll tell you about that. But one of the main reasons was that if you think about evolution and natural selection, once you age, you've probably already had your children. So even if you age in a better way, it's too late to pass the good genes on to your children. So that was the idea of why there couldn't be genes that controlled aging. So basically, there wasn't really any research done. Meanwhile, people were finding cancer genes and genes for all sorts of other interesting aspects of biology, but nothing for aging. Okay, so I want to talk now about a little history of how this paradigm really changed dramatically and in a wonderful way. Okay, so I had something to do with this. First of all, well, and then I'll go to the history before me in a second, but my getting into this had to do with the fact that I studied developmental biology. And often when you study one thing and then you look at something else, you carry the ideas that you get from studying the first thing to the second. And what I discovered in my work on this little worm that you're going to learn a lot about in just a second, as a postdoc, and when I was first at UCSF as a professor, was that they had in their bodies a set of genes called Hox genes that patterned the body axis all from head to tail. Now, these were already known to be present in fruit flies and mice and people. And so it's really interesting because flies and mice and worms look completely different. So you would think that everything that makes a fly look the way it does would be totally different from what makes a worm look the way it does. But no, they have these regulatory proteins. There's transcription factors that control gene expression patterns that are the same. In fact, you can take this gene, one of these genes, out of the worm, and you can stick the fly gene in, and it works just fine. It makes a worm pattern, not a fly pattern, because the downstream genes are worm genes, but it works just fine. So that was really important. There are universal pattern regulators, and who would have ever thought that that would be the case? But it is the case. So if you get that in your mind, and then you think about aging, I won't say, but what comes to your mind? a universal aging regulator, obviously. So it might not be the case, but it might be the case. So I had this idea in my mind just based on what we had learned in other people too from studying development. So we studied this in C. elegans. Now I want to introduce you, this is my little friend, C. elegans. It's an old one, but they're very small. They're about the size of a comma in a sentence, and they age really quickly. In just about 2 weeks, they're old and dead. So it's pretty nice to do experiments in these worms. And actually, they have all the different cell types and neurotransmitters and lots of pathways that we have in our bodies that control important things like cell division and death and muscle function and all that sort of stuff, neurotransmitters. Okay, so we wanted to study aging, and we wanted to study it in C. elegans, and it helped that we were already working on C. elegans. Now, at the time, there had already been a long-lived mutant reported by another lab called Tom Johnson's lab. And nobody knew anything about it, but the worms lived long. However, they didn't reproduce very well. So there were other evolutionary models having to do with the idea that if you don't reproduce well, your resources could go toward longevity. And so it wasn't really clear how interesting this was, but there it was. It was a long-lived mutant. So, okay, so the first thing we did was we took normal worms and we took out their reproductive systems with a laser, so they couldn't have any children, and they had a normal lifespan. So that told us that aging there wasn't some kind of simple trade-off like that. And then we looked for mutants. And actually, this is interesting, and if any of you are students, hopefully some of you are, I think it's really interesting because everyone, at the time, remember, there was nothing to control aging. That was really the truth, quote. And if you thought that differently, people thought you were crazy. And they would really look down on you in a scornful way. And so that happened to me. It was really hard to get the project started. Nobody in the lab, they laughed at me, they put things up on the board. I was in skits like Cynthia doesn't want to get old, so she wants to study aging, which was actually true. But anyway, but finally we did. I was taught to rotation students and not let them go anywhere near my graduate students. Rotation students would just spend a few months in the lab before they decided to join or not. And finally one said yes. And so then we did so the moral here, you'll see there was a good ending to this, that if you think that you have a good idea and you really carefully criticize that idea and you can't find anything wrong with it, then even if people hate it, it actually might be a really good idea, because if it's true, then you might change the way people think, which is a good thing. And so I think then you should go for it, if you can. So we set out to do a screen, and we were very lucky to find a long-lived worm. And this is a movie. C. elegans, beautiful worm, moving along. That's normal worm. And in just 2 weeks, look, get your handkerchiefs out. This worm is barely moving its head at all. And you can see it's old. See what I mean? How universal old. This is the long-lived mutant at the same age. Okay, those guys lying there in the nursing home, these guys just moving around. So it's not a miracle because it was science, but it was like a miracle. You'd see these 2 plates of worms, everybody's almost dead or already dead, and the other's just moving around, and you think, oh my God, what have I done? But I've done a good thing. And I said to myself, I want to be those worms, and that's why I'm trying to do that now in a company, actually. All right, so onward here. Okay, so here is the lifespan of a normal worm, black. Here is the lifespan of the DAF2 mutant, twice as long. And we knew already that the DAF2 gene was a regulatory gene. It controlled a set of decisions made during development. And in order to do its thing during development, it needed another gene called DAF16. So we asked, does it need DAF16 for this long lifespan? And the answer was yes. So that told us not just that you could change the lifespan dramatically, but that it was actually subject to regulation, because regulatory genes were controlling it. Okay, so what are these genes? DAF2, this one here, encodes a hormone receptor. Again, this was shown by Gary Ruvkin's lab several years later, similar to a receptor that we have called the insulin receptor and also the IGF-1 receptor. These receptors are in all animals. They're essential. But what we did here in this mutant, they're essential in the worm too. Like if you knock it out completely, worms don't grow. But if you just make it not work quite as well, then you get this amazing long lifespan. So the animal has within it the ability to live twice as long as it does. Because we just changed one base change in a hormone receptor. We didn't really get in there and tinker with all the nuts and bolts of aging. But we doubled the lifespan. So it had the ability to do it. We just had to coax it out. And the DAF16, our lab and also Gary's lab showed that it encodes a transcription factor. And if that isn't regulation, then what is? Transcription factors they turn genes on and off. So that said that the animal is really reprogramming itself to change its lifespan. Okay, a big picture. I don't think that we would have invented airplanes if we hadn't seen birds fly. I mean, who would think that you could put an object up in the air and have it just stay there unless it was a cloud? But there were birds, heavy birds, that could fly around. And I really think that was why we thought maybe we could make an airplane. And I think with humans and with animals, all animals age. There aren't people that live to be 200. So but some people get cancer and some don't. So you think, well, maybe we could do something about cancer because you don't have to get it. But everybody gets aging. So people think, and with the evolutionary biologists with their ideas, then it's not even worth studying. But here you have a situation where you have genes controlling aging and switches that molecular biologists know about, like hormones and kinase signaling pathways and transcription factors. So you can study it. It's molecular biology, and maybe you could live longer, maybe, and better. These animals are really healthy, and they're also disease resistant too. Okay. So just a little picture here. The partially active receptor, it's interpreted, I think, as a danger signal. These receptors respond to cues from the environment saying that there are lots of nutrients and things like that. And so stress, if you don't have a good receptor, maybe the animal thinks it doesn't have enough food or it's too hot, something like that. It has too many pathogens. And what happens is a whole collection of stress-responsive transcription factors, like this one, DAF16, which is a FOXO family transcription factor. And these famous ones, heat shock factor, NRF2, TFEB, which regulates autophagy, and even more, they all go charging into the nucleus and they reprogram the cell to make the animals more resistant to all sorts of stresses, including, I think, the stress of aging. And that's what makes them live longer. Okay? And actually, so this brings us back to evolution. Okay? Maybe it can bring me back to, yes, implications for evolution. So this is really cool. Remember I told you the evolutionary biologists, they said, okay, you could never have a kind of a gene regulatory system for aging because it would only affect you after you reproduced already. But this pathway and system, it makes you resistant to all sorts of horrible things like basically almost anything, like bad salt concentrations, a high temperature, low calories, many, many, many different stresses, even when you're young, even when you're a teenager, before you reproduced. So that means that this regulatory system is a very good reason for it to exist. If it's there, you might live long enough to reproduce. But then it's still there. You lived and you already reproduced, but there it is. It's still there in you. And now it's controlling your lifespan. See? So there is a way for this to evolve. Not that it did evolve this way, but there's a perfectly plausible explanation for it. Okay, I'm not going to talk about this in any detail, but basically there's all sorts of ways that you can get DAF16 to go into the nucleus and extend lifespan. You can do it by caloric changes, sensory cues, heat, energy sensors, even timers, which I won't go into right now. But the important point of this is that I don't think there's just one way to activate the system to extend lifespan. There's probably many ways. Okay, actually, and if you do two of these things in the same animal, which I'm not going to go into the details, you get this unbelievable lifespan extension. So here's normal worms in black. Here's the famous DAF2 mutant. And here is when you do both of those things together. Now, these worms were photographed when they were 144 days old. Okay, so they were here. Now, at that time, normal worms had been dead this long. So they were, like, they'd be eaten by then. You wouldn't even be there anymore. But these guys are still moving around. So lifespan is very, very plastic. And that makes sense because of evolution, right? A common precursor to us also gave rise to the worms, which have a very different lifespan. Okay. Now, other species. So you change these genes, flies live long, mice live long. And actually, these are among the longest-lived mice that there are, mice mutants that have been described right now. Here is a picture of the regulatory circuit. Oh yeah, they have lower rates of cancer, fewer diseases, fewer senescent cells. Okay, and now, this is just a bunch of arrows pointing to different tissues. This is a picture of the regulatory system in a mammal that controls the release of IGF-1. And all these are all genes, essentially, that play a role in this pathway. And the red arrows point to ones where if you change them, the mouse lives longer. Okay, so that's pretty cool, and we have all these same proteins in us. Okay, what about, could this affect evolution? Well, if you went to Vera's talk, you heard that some bats live a very long time, and they appear to have very defective growth hormone IGF-1 signaling, suggesting that, we don't know, no one's done the experiment, but it suggests that maybe changes in this system in evolution can account for at least some of the changes in lifespans that we see around us. And small dogs, little dogs, they're actually mutant in IGF-1, so they're small, but they also live a lot longer. And we know that you don't have to be small to live longer because in experimental animals, you can change the pathway only in adulthood, and you still get the life extension. So there are two very familiar cases. Ponies are similar, small horses. Okay, also people. So like I said, we don't have people that live to be 200s. We don't really know about that. That would be really interesting, but we don't have them. However, when you look for genes that are overrepresented in people who live a long time, you do find variants affecting IGF-1 and also FOXO3, which is DAF16. And I'll come back to that in just a second. Also, I should tell you that Raptor, which you heard about just now, is another gene in this network. And inhibiting TOR extends lifespan. And you can inhibit TOR with a drug. So that the point to that is that it's a nice example. People can take this drug. It has side effects, so it's not really what you want to take for lifespan. But it shows you for sure that you can drug some of these guys and get effects on lifespan in this system. Okay, so we wanted to, so I'm at CALICO, which is a Google-associated company that is focused on aging and the basic research of aging, but also trying to translate some of this into, trying to make our health spans better, give us a better life when we're old. Okay, so we've been looking for more genes that might affect lifespan in humans. So the thing is, all humans, we have different genes. That's why we look different. We have different forms of our genes. But we all have pretty much the same gene complement when it comes to aging. We're not that different. Everybody has about the same rate of aging and lives about the same. We don't have people that are really healthy that go around and age the way dogs do. There are progeric people, but they don't really look like real aging. So when you try to find genes but the hope is that there might be some genes like DAF2 in humans that differ just a little bit, but enough that you can see it. So maybe in the worm, maybe we change the gene activity by half, and so we get this big effect. Or maybe in humans, a similar gene, maybe it's only changed by 5%. So you don't get this big effect, but you might get a little effect that maybe you could see. And then if you find some new genes like that, you could investigate them using mice and other organisms, and humans also. And then maybe those would be nice targets, and you would start from humans, which is nice because we're humans, and so then you would be pretty sure that it would affect us. Okay, so we looked for genes affecting aging in humans using the UK Biobank. So the UK Biobank is a wonderful resource in the United Kingdom. That's why it's called UK, where they have about 500,000 people, and they phenotype them for all sorts of things, many, many different phenotypes, hundreds, like their grip strength and all sorts of things. Some of them are listed here. And they also have some genotype information, some information about their DNA. So we wanted to find, so we thought to ourselves, okay, among those 500,000 people, maybe some are aging slowly. So no matter how old you are, people your same age, and probably some are aging really fast, and some are aging really slowly. And that's interesting, because aging is time dependent, but it's not always the same rate. So the question is, can we find people who age more slowly from the UK Biobank, and then can we see if they have genes in common? Okay, so that's what we tried to do. Serge Leibert led the charge here. He's brilliant, and he's working in my group at CALICO. So here's some examples of phenotypes that change with age. How long it takes you to tell if two pictures are the same, latency to a match, your metabolic rate, your grip strength, your blood pressure, and there's a whole bunch of other ones. So we took about, I guess, over 100 of these phenotypes, and 113. And we used principal component analysis to combine them in such a way that you could best predict a person's actual passport age, their chronological age. I won't go into the details, but it was done completely mathematically using these as inputs. And here's what we come out with. So here's, we come out with a line. This is people who, and these are people. These are the 500,000 people in the UK Biobank, and each dot is a person. You would be here somewhere. If you're up here, that's terrific, because you're young for your age, and etc. Most people are right on the line, and there are fewer people out here. So one thing that's interesting, so we wondered, is this just noise, or is it meaningful? Well, it turns out these little black dots here are people who died within 5 years of being in the UK Biobank. And it turns out that you're more likely to die if you are old for your age. So here's another way to plot that. So here's delta age. This is the distribution of people. Delta age means that how far you are biologically from your chronological age. So a delta age of 1 year would be 1 year older or younger. So anyway, here we are. This is our distribution of people. And here in red are the people that died. And what you can see is that that curve is shifted over here. So if you have a young delta age, so you're young for your age, then you're less likely to die. And if you are old for your age, you're more likely to die. And interestingly, you can do these mortality rate analysis on this, just like I showed you with the Gompertz curves at the beginning of the talk. And it turns out that each year of delta age is about 8%, which is about the rate of change in your risk of death per year. So so we think it's real. There's a lot of other reasons, like it predicts your parents' age at death and other things like that. So we did GWAS. Here's FOXO3. It's there, we saw it. That's DAF16. It comes up in a lot of GWASs. And we got a lot of other loci that come up when you do GWAS for people who live for a very long time. I haven't labeled all these here. This is on bioRxiv, the paper, so you can go and get the lists. Here's the actual list of the genes. And I'm not going to talk about them because I don't have time. One thing we did that was kind of interesting is we asked, okay, if you want a gene that affects all of aging, then no matter what part, if you subtract away any of these subsets of phenotypes, you should still have an effect on aging. And it turns out we hierarchically clustered all these traits together, like the left hand and the right hand grip strength are very similar to one another. So they're very close together in a cluster. And it turns out that these clusters, more or less, we colored them in. Now, the clustering was made mathematically, but we colored them in subjectively. So these colors and these little things here, we just put them because when we looked at this, we thought, there's a lot of muscle-specific traits here, cognitive traits here, cardiovascular traits here, and so forth. And so then what we did was we redid our GWAS, taking out one by one, 12 times, taking out one of these clusters. So like if we took out, say, for example, the kidney cluster here, the thing that we call the kidney cluster, then if one of those GWAS hits affects your biological age only because it affects the kidney, then it won't be there anymore. And so we did that. And when we did that, about half of those genes that were in that list went away. So those are actually interesting because they may be genes that are new genes that have a function in, say, kidney disease or something like that. But they're not really good targets for all of aging because, so the ones that are left are good targets. And we also did something else. Some of the people in the UK Biobank have checked a box saying that they have a disease, like they have cancer or they have had cancer or something like that. So we took all those people out. We ended up with about half as many people. And again, a lot of these GWAS hits went away. And when you look at them, you can see why. Some of them were very, very disease-specific GWAS hits. But again, some stayed behind. So those are the ones that we're kind of combing through to get ideas about follow-up. Okay. So I'm just going to say a few more. Just a few little things I want to say. ApoE, famous risk allele for Alzheimer's disease. If you have ApoE4, if you're a homozygous ApoE4, then you're very likely to get Alzheimer's disease. And if you're ApoE2 homozygote, you're very unlikely. And if you're 3, you're in the middle. So because people who have ApoE4 alleles are likely to die, when you do longevity studies, well, it's not surprising that you see those people, because if they have Alzheimer's and they're only 80 or so years old, you won't see them. They won't be centenarians. So it could be that they're not there, that it comes up so frequently in GWAS hits because it affects your ability to live to a very long time. But what we found was really interesting, was that no matter how old you are, if you have the E4 allele, you have an older biological age, and if you have the E2 allele, then you have a younger biological age. So it's really interesting. And when you take out any one of those little pie slices, it never goes away. So it's doing, and I won't go into it in more detail, but that was pretty interesting to me. And then we did gene ontology analysis on the different GWAS hits to see, was there a certain function that stood out? And the one that stood out 18-fold over anything else was your synaptic function. There's a whole list of genes that are like synaptotagmin. I mean, they're very, very specifically involved in synapse function. And those genes were very highly overrepresented in this set. And then we looked to see whether any other traits that weren't actually in the model, that were kind of behavioral traits, correlated. And what we found, which was kind of interesting, is that people who play computer games are more likely to have a low delta age. And this is it's really hypothesis suggesting more than anything. But there it is, there's the data. And finally, I'll just say that we took 110 or so phenotypes, like grip strength and heart pressure and things like that, blood pressure. And we put them together, the model put it together, principal component analysis did. It asked which of all these traits, when you combine them, can predict biological age the best, chronological age the best. And from that, so we analyzed those traits, and we found that there were only about 12 or 13, some for males and some for females, that some were the same, and the ones in color were sex-specific, that you could get about 90% as good result if you used only 13 or so traits as compared to all 110 traits. So these traits are things that you could imagine you could measure in a clinical trial to see whether something is making you younger. Like you take a pill and you ask, okay, am I younger? You eat seaweed, we ask, am I younger? There's a lot of other things like the methylation clock, et cetera, but this is a nice new index that's simply based on mathematics that one could consider using as well. Okay, I hope I didn't go over. I have no idea how long I took, but these are people whose work, many people have worked on aging in my lab for many years, and they're great. And I highlighted the people's work who are shown here. And then this is a picture of Serge, and he's with me at CALICO. And we're studying humans and worms and mice and all sorts of things, hoping to slow down aging. So that's it, and I'll take questions. And so will Henry.
Thank you so much, Cynthia Kenyon. We will now take questions. So we will take questions from the audience, and we would like to start with you, Dr. Kenyon. To what extent could a human being actually calculate its biological age?
Well, if you happen to be in the UK Biobank, you can see which dot you are. And then that will tell you. If you're chronologically 30, but you have a delta age of -5, then your biological age is 25. Now, I can't actually, there's a lot of scatter in those plots, so you could see that. So it doesn't really mean all by itself a whole lot. You can probably tell just as much by looking in the mirror. But at a population level, I think it is very significant. And that's one way you could have your methylation. You're going to hear about this really cool methylation clock, which is a molecular clock in this afternoon. And that's another way you can do it. So how accurate is it? I would say at the individual level, it's good to know. It's good to know, but I don't think I would take out insurance policies based on that myself. Okay.
Perfect. Thank you. Dr. Jasper,
Yes.
will gene therapy ever be a standard practice? What do you think?
I think it's hard to predict this. There are many safety concerns, and this is one of the hardest parts of drug development, is to try and make sure that any interventions that we believe are going to be useful are deployed in a safe way, right? However, there are examples now where gene therapy is being used. There are examples in the retina, for example, where viral delivery of a gene that is missing in certain patients that lead to retinal degeneration is being resupplied and has real impact and has been approved by the FDA as a therapy to be used in these cases. So I think as we learn more about the opportunities and the safety of these strategies, we will see more of these gene therapies come to market and be accessible to target very difficult diseases. How widespread it will be is a very hard prediction, and we'll see. But we are in an exciting time right now.
Perfect. Thank you so much. Back to you, Dr. Cynthia Kenyon. What do you believe are the next challenges to be investigated regarding the slowing down of aging?
Well, as you've heard from these talks, there's a whole lot of ways you can slow down aging in mice. Mice are very lucky animals. And really a lot. And they seem different. They may all be connected in some way, but at least superficially they seem like different ways. So I think you'd have to be a pessimist. I mean, we are animals also. We share a common ancestor with mice. And we know a lot about our biochemistry and our functions, and at the molecular level, a lot is really, even though we don't look like a mouse, we're pretty similar in many ways. So I think you'd have to be a real pessimist to say that none of those will work in humans at all. I think it could be, but I think you have to be pretty pessimistic. So what you want to do is you have to try a lot of things, and that's what we're going to try. The good news, we're really lucky because it's really hard to do a clinical trial for aging. In the US, it's not even approved that you can do that. But you can do clinical trials for diseases. And what's really great is, as you know, age-related diseases are related to aging. So if you push back aging, you should have some effects on diseases. And as a matter of fact, all those worms and flies and mice I talked about, if you cause them to get a disease, they have either no symptoms or fewer symptoms. So they're protected against a lot of diseases. So the idea would be that you could take an intervention that you might hope would slow down your aging rate, get it approved because it affects heart attack frequency, something like that, for example. And then people are taking the pill and it's good for them. And then you can ask, if they're not too sick, you can measure aspects of aging, their methylation clocks, other aspects, and see if they're aging more slowly. So it's already, then people become kind of your experimental organism for the drug. So I think we might get lucky and the first one might work really well. Maybe we can make rapamycin safer and that will work. I don't know. Maybe not. But there's a lot of things we can try. And I think that what's really nice now is I think finally people are getting it. They're getting the idea that aging rates are not set in stone. They can change. And maybe they could change in humans. And let's go for it. Let's try it. So I think there'll be a lot of trials. And I hope there'll be some good successes. I believe there will. And I really hope that is true.
Let's hope so. Thank you.
Yeah.
Dr. Jasper, to finish off, do you believe that the rejuvenation will one day be accessible and available for everyone? Let's say everyone that's sitting in the audience right now.
Well, I think if you just heard what Cynthia said, there's certainly an opportunity here that some of the interventions that have been described, which often are nutritional interventions, exercise and so on, which really lead to an impact on health span and lifespan in other organisms, will also apply for humans. So that by itself, more exercise, for example, is a very accessible thing. When we talk about really more targeted and complex interventions, for example, gene therapies for reprogramming, rejuvenation, and so on, the biggest part of the work will be to make these work and make them safe. That's the biggest part, and that will take time. As we develop these drugs, the question of accessibility is ultimately then a societal question, right? It's a question about healthcare systems, it's a question about financial viability, drug costs, etc. And this is really something that, as a scientist, I'm not qualified to answer. But we all know that there's more work to be done to really make these things accessible. But the very first step is going to be to make it work and to make it safe. And that's something we're working on. It's going to take a while, but there is a lot of promise there. And I'm as optimistic as Cynthia is that these things, that what we've learned over the last 20, 30 years is really something that helps us target maybe not aging itself, but at the very least age-related diseases and have a real impact there.
Thank you so much. Unfortunately, we do not have time for any more questions, but I would like to thank the audience for joining us in this 5th session of Age Open Science. Our next meeting will take place at the claustro of the Colegio Arzobispo Fonseca, which will start shortly. I please remind you a los asistentes que por favor depositen el dispositivo de traducción simultánea a la salida, and to our speakers, it has been a pleasure to have you. Thank you so much.